Just a quick recap first of all. Hopefully we'll remember that when we are talking about prokaryotic cells, these are things like bacteria. So when we talk about the bacteria, these are prokaryotes, and therefore they are made of prokaryotic cells. Hopefully that is pretty logical, and we all knew that first point. When it comes to what these prokaryotic cells look like, this is a typical diagram of a prokaryotic cell. Now, what we will actually find is that while we have a range of structures on this, not every prokaryotic cell will have all of those features. So there are some that are only present in certain prokaryotic cells, but this gives us a good overview of all those different bits that do exist. So what we can see, we do have a cell wall, just like obviously in our eukaryotic cells of some cases. We do have ribosomes that are present, there is cytoplasm, we have a cell membrane. So we do have some of those similarities that we can pick out from the diagram alone. But there are also some new bits on here that we've not previously encountered. Things like pili, for example. We've never mentioned pili before. If we start off with the similarities then to those eukaryotic cells, what we've got are basically these four key things to remember. They have a plasma membrane, just like our eukaryotic cells have a plasma membrane. They have cytoplasm, they have DNA and RNA that's present, and they've got ribosomes. So those are our four key similarities. When we come on to the differences, some of these should be familiar to us from our GCSE work, but some of them will be obviously new for the A level. So first of all, hopefully the familiar one is that these are smaller cells than eukaryotic cells. When we come on to some of our new bits, they're less well developed in terms of their cytoskeleton, and they don't have centrioles. So key distinction there between our eukaryotic and our prokaryotic cells. Prokaryotes, less well developed cytoskeleton with no centrioles, whereas the eukaryotic cells obviously have a very well developed cytoskeleton, and they do have those centrioles present. Again, a GCSE throwback, there is no nucleus in a prokaryotic cell. Eukaryotic cells all have a nucleus. If you're looking for one difference to remember, remember that one because it's a good old favourite. And they have no membrane-bound organelles. If you were asked for two differences between prokaryotic and eukaryotic cells, do go careful. If you were to try to put down they have no nucleus and they have no membrane-bound organelles, technically the nucleus is a membrane-bound organelle. So you could very well be putting down the same marking point, just two slightly different ways. And that means you would only get one mark, not two. So always try to go for two rather distinct features rather than ones that have that overlap, like no nucleus, no membrane-bound organelles. Pick one of those and then go for something else as well. We do have a cell wall which is similar to the obviously plant cells and our fungal cells. However, the cell wall within our prokaryotic cells is made of this substance called peptidoglycan. So plant cells, cellulose, fungal cells, chitin, and our prokaryotic cells is peptidoglycan. So make sure you know the components of those three cell walls in those three different organisms. They also, in prokaryotic cells, have smaller ribosomes. So while they have ribosomes, the ones that we find within prokaryotic cells are smaller, and we call them 70S ribosomes. The ones within our eukaryotic cells are what we refer to as 80S ribosomes. And then we also, rather than having our DNA contained within the nucleus wrapped around histones as we see within our eukaryotic cells, in our prokaryotes, it's a naked loop of DNA, so that means it's not wound around those histone proteins, and it's just free floating within the cytoplasm, so it's not contained within that nucleus. As we said at the very beginning of this little summary, even though on that diagram we have a whole range of these features, not all of them are present in all prokaryotic cells. So we've got four little structures here that are only present in some prokaryotic cells. we would have a waxy capsule for some of our prokaryotic cells, and the whole purpose of this is to act as basically a protective capsule around it. So it protects it from things like drying out, for example. We will also find plasmids. Now, the plasmid is a little loop of DNA, so usually a little circle is how they represent it in diagrams. And this will contain additional genes, and quite often things like antibiotic resistance genes are found on those plasmids. We'll also find flagella. Now, the flagella are those pink things on the diagram there, and those are all to do with movement. Because when we're thinking about these prokaryotic cells, they don't just want to be just stuck in one place. In some situations, they need to be able to move. So if they're in aquatic environments, for example, then having the ability to move through the water, for example, is going to be very beneficial to them because they can relocate to a more suitable area. And they've got these things called pili, which are these small hair-like projections around the surface. They're the little green things on the diagram there. And these are used for, first of all, adhesion. So by that, what we mean is that they can stick to surfaces, and they can also be used to transfer those plasmids from one bacterium to another. So basically it allows two bacterial cells to join together in a way, and then they can transfer the plasmids from one to another. That does mean that they can then transfer those all-important genes, like the antibiotic resistance genes, from one bacterial cell to another bacterial cell. When it comes to how our prokaryotic cells divide then, this is a different process to what we see in eukaryotes as well. So the process by which our prokaryotic cells divide is binary fission. If we look at those words, then binary, so hopefully we remember that bi basically means two, and fission is all to do with splitting. So basically what we're doing here is we start off with our first cell, and then we're going to split it to make two. So we start with one and we make two at the end. So the way that this is going to happen, obviously before we can just split a cell into two, we have to copy the key bits, the DNA and the plasmids. They have to be copied prior to the cell dividing. So just remember, binary fission is the way that we phrase cell division for our prokaryotic cells. One thing that's really quite useful to help you get your head around all of these kind of key features of both prokaryotic and our eukaryotic cells is to create a little summary table. Now, I've created you a lovely summary table here that you can obviously utilize. So what we can see, some key features in that first column, prokaryotic cells in the middle, and then the eukaryotic cells on the right. So we can see the difference in their sizes there. So eukaryotic cells obviously much larger there, 10 to 100 micrometers, whereas our prokaryotic cells 0.2 to 2 micrometers. Membrane-bound organelles are absent in prokaryotic cells, but present in our eukaryotic cells. The different material that we find the cell wall is made from, peptidoglycan in prokaryotic, whereas in our eukaryotic cell, depends on which one we're talking about. If it's plants, it's cellulose. If it's fungi, it's chitin. The cytoplasm, obviously we find a less well-developed cytoskeleton in our prokaryotes, whereas in the eukaryotic cells, our cytoskeleton has those kind of four key components that we looked at in our previous video. So the microfilaments, the intermediate filaments, the microtubules, and those motor proteins, all having very important roles there. In terms of the ribosomes, prokaryotes are smaller. So remember, we could phrase that as their 70S, and the eukaryotic cells are the larger ones, or 80S. The DNA then, prokaryotes, single loop with no histones. Eukaryotic, multiple linear chromosomes with the histones associated with them. And finally, the process through which the cells will divide, binary fission for prokaryotes and mitosis for our eukaryotic cells. One of the things I'd suggest you do is basically draw out the outline of this little table and then just practice filling it in from memory. So the idea being that you just have the blank outline, you can then try and fill out as much as you can, then double check it. Once you've done that, obviously make sure you've got all the ones right that you managed to fill in, review the ones that you didn't remember, and then just try it again. If you repeat that over an extended period, obviously not just sit there for 6 hours doing the same thing, but if you do it one night, then obviously come back a bit later on a different night and try again, you repeat that over a longer period, what you're going to find is this will stick. And rather than just being in your short-term memory, you're going to have it in that long-term memory so you can draw upon it when it comes to those exams at the end of year 13. As always, don't forget to subscribe to the channel so that you will always be notified of any of our new updates as I then upload some more of our A-level biology videos.